Hi, good afternoon. I'm here in Estoril, Portugal in the ESH Translational Hematology meeting. And the question I was asked to answer was whether we can predict AML with genetic testing. So the short answer is that we are in the process of being able to do that. We can now offer healthy individuals that we might be at risk based on specific blood tests to undergo a specific molecular testing, which is deep sequencing of targeted and specific genes. And based on the results of the sequencing methods, based on the results of the sequencing, we will be able to define high-risk populations based on several parameters. One of them is the type of mutation, so the specific type of mutation. The second is how many mutations a healthy individual carry. The third is how big is the size of the clone, or how many cells actually carry the mutations, and a few other parameters, and we take them all together into a mathematical model that can predict whether a healthy individual will have AML in the next 8 years. Currently, we cannot take this into practice. We cannot offer these tests to the general population, as AML is a rare disease and we still have some false positive in our testing. However, we believe that we will improve our predictive efforts, and we will find parameters that can be applied to the general population, like the increased RDW that we already identified as a risk for AML, which is a routine test that almost every individual does. So based on routine tests, we'll be able to define a high-risk population that they will be offered to undergo genetic testing to predict AML. The issue that everyone asks is whether we can do anything with this knowledge. So let's say we know that someone is going to develop AML in the next 8 years. So we believe that clinical trials should be designed to actually target the individuals with specific mutations if there are targeted therapies like IDH1 and IDH2 inhibitors. So individuals carrying these mutations can be enrolled to preventive therapy, and the outcome shouldn't be the prevention itself, because that can take many years. We should monitor biomarkers like the size of the clone, and if it reduces under the treatment, that means that we might be either delaying or preventing the disease, which might be good enough. Not totally eliminating the disease, but delaying it to a long enough period so other diseases will come and not AML. So I think this is in a nutshell where we are now in the early diagnosis and prevention of AML. Thank you.